science. I'm doing science. I'm doing science. Science time.
Hello, my weird and wonderful human beings, and yes, you heard that right. I am doing science in a lab with actual other scientists. Can you tell I'm excited? The joys of grad school is that, well, as an undergrad, you mostly get to learn about these cool theoretical models and occasionally touch an actual microscope. As a grad student, not only do you get to find areas of science that you're interested in and then focus exclusively on exploring those, but you actually get to have a hands-on role in creating, conducting, and analyzing your own research. And in case you can't tell,
I'm just a little bit excited.
for any of you wondering where I have been, that is where I have been. In a lab, doing actual science. For those who are new here, hello, this probably seems like a lot. My name is Eve Daniels, and I am a grad student studying experimental and medical bioscience at Linköping University here in Sweden. And as I am now entering the second half of my first year,
I get to science.
I've mentioned before how when you go to grad school, you get to pick a research project and then research it. But I haven't explained sort of exactly what that means. All grad schools are different, but generally speaking, when you're in any kind of graduate program for the sciences, you have to find, conduct, analyze, and then present a research project. Different programs have different restrictions. I know some schools want you to work only with faculty. Other schools prefer that you focus in a specific area, especially if that's an area that they're really invested in. But at LAU, the world really is your oyster. I know classmates who are working with industries and outside research facilities. A couple of them are even working with the police department, which is pretty cool.
Very CSI. And yes, like all scientists, they hate CSI. Don't at me. It's garbage science, terrible lab techniques, and they can't keep the lights on? Why is that lab perpetually half lit? I mean, really.
A research project is a really big part of the whole grad school thing. And it's a part that can actually shape your career and the fields that you go into. So finding or choosing a project can be kind of stressful. Now, this is something that I talked about in the fall when I actually found my research project, but I didn't go too much into the details, partially because the details for my experiment hadn't been finalized and we weren't actually sure what my experimental procedure was going to be, but also because I wasn't really sure what I was allowed to discuss. Part of ongoing research means that you're dealing with unpublished data, new discoveries, things that have patents pending, and just generally you don't go blabbing about stuff like that. Plus, since I didn't have a finalized project at that time, there wasn't actually much to share. But now that I've actually been in the lab for a couple of weeks at this point, let's talk science. Most science sort of evolves as you go, and we're no exception, but we have a general idea of the plan. I'm going to be working with Maria Lerm and her research group at LAU. Maria is a professor and researcher in the Department of Biomedical and Clinical Sciences in the Division of Inflammation and Infection.
And just before anyone comments, I'm not being disrespectful by referring to my professors by their first names. That's just how it works here in Sweden. They have a very lateral sort of organizational system. It's not a respect thing, it's not a rank thing, it's just a cultural thing.
So the research that we're actually doing relates to epigenetic memory and how that relates to the immune system in combating certain diseases. Specifically, we're looking at how changes to certain cells in the human immune system can actually make those cells better at fighting things like Mycobacterium tuberculosis, also called TB.
I know that's kind of a lot, but I'll explain.
Basically, all of my research starts here, with this article. This study discovered a link between altered methylation patterns in immune cells and increased antimycobacterial activity. But what the heck is that supposed to mean? Basically, your immune cells usually fight off invading pathogens in one of a few ways. The one that you are probably most familiar with is called phagocytosis. It's where your own immune cells reach out and grab onto invading bacteria or viruses and basically eat them, Pac-Man style. But this doesn't always work. In some cases, the invading bacteria is just too big for your immune cells to eat. In which case, your immune cells have to resort to slightly more drastic measures. Your immune cells will essentially self-destruct, setting off a chain reaction that tells other nearby immune cells to also self-destruct. But how does your immune cells blowing themselves up actually help you fight off bacteria? This is the cool part. The leftover DNA and bits of cell membrane that are still floating around will actually migrate to surround the invading pathogen, creating a net. Once that invading pathogen has been corralled, they can work together, even though they've already self-destructed, to kill the invading pathogen. Sometimes with a little help from other immune cells, but it gets the job done. The question is, how the heck do they pull that off? And basically, that's what I'm trying to figure out. This is a really interesting field because it has so many different directions to go in.
We could be looking at pathology, we could be looking at host immune systems, we could be looking at epigenetics and the idea of genetic memory in general.
There's also a lot of bioinformatics involved, which is a whole separate conversation. Not only is tuberculosis a fairly serious global health risk, particularly in more impoverished nations, but the things that we learn by studying host immunity and the immune system reactions to TB could lead to novel treatments in other diseases within the Mycobacterium family and even other diseases in general. So far, most of what I've done in the lab is math. But I have actually been able to start some of the practical bits this week, which is why I've been kind of radio silent lately.
In the lab, play with my cells, my little test tubes.
And most of what I've been doing so far is pipetting clear liquids from one tube into another tube of clear liquids.
Science.
I did that for 5 hours yesterday, which explains why my back looks like this. Those clear liquids that I put into other clear liquids that we put into other things are eventually going to be treated with human immune cells and tuberculosis to examine how the two interact using a very novel piece of technology. I will link a video down below in case you're curious. And I do hope to be able to take you guys on sort of a lab tour sooner or later, but I need to make sure that that's okay first. Maria is amazing. I gushed about her when I found this project because she's always just so on the ball and really responsive. She has been so supportive in helping me fill the gaps in my own knowledge. It's no secret that when I started grad school, I didn't have a ton of lab experience, as in the last time I had set foot in the lab was almost half a decade ago. But Maria just took all of that in stride and worked with me to help figure out ways that I can fill my own knowledge gaps. If I have a question and she doesn't have time or she doesn't have the answer, she knows exactly who I should ask. For the record,
Future scientists, if you're confused about what a piece of equipment does, there is a YouTube video about it. Usually created by the manufacturer. See my lab techniques playlist.
more than that, the research that we're conducting in Maria's lab is part of a collaborative effort. There are a half a dozen researchers at any given point working on this same subject matter in different capacities. One person may be working on gene silencing or sequencing or splicing. Another person is looking at antibiotics and novel drug treatments. Somebody else is dealing with bioinformatics. And you have so many different backgrounds and so many different depths of experiences coming together that no matter what you want to figure out or what field of expertise you may not be familiar with, there's someone else there in the group to offset that. We also have Zoom meetings twice a week on Mondays and Fridays, which has been a lifesaver for me. We get to go over new research, troubleshoot ongoing experiments, and it really helps everything to feel like a collaborative experiment and for us to feel more connected to each other and other ongoing projects, especially because due to COVID restrictions, not everyone's in the lab right now. And this has actually been a big problem for some of my fellow classmates. Finding projects with researchers who aren't necessarily in the lab all the time or who require more supervision. I know people in my class who spent months waiting for responses from researchers. Some of them did eventually get a response, and some didn't. So that's me. That's what I've been up to, and that's what I will be up to for the next 4 months. Fingers crossed that my pipettes remain accurate, my cells explode magnificently upon exposure to tuberculosis, and that my spine survives pipetting 384 well plates multiple times a week. If you guys have any questions about sort of the research that I'm doing, or grad school in general, or grad school in Sweden, make sure you comment them down below, and sooner or later I will collect a bunch of them up and do a little Q&A. You can also hit me up on my Instagram, where I don't always remember to post, but when I do, it's normally because I'm doing something pretty cool. I hope you guys are safe and well, and that wherever you are in the world, you keep learning, keep growing, and stay curious. So now, we science. Can you tell I'm excited?